Welcome to the deep dive. Today we're jumping right into a really big reorganization that's happened in the world of microbes. We're
Oh,
talking about Lactobacilli, that huge group of bacteria.
yeah. And these aren't just, some obscure things in a lab. These are the real workhorses behind foods you probably eat all the time. Yogurt, cheese, sauerkraut, pickles, sourdough bread. They even play a role in making chocolate and wine taste right.
Oh, definitely. And they're also super important for our health, right? For our gut and animal guts too.
Absolutely crucial players there. So you're encountering them constantly, maybe without even realizing it.
Right. But here's the thing. For the scientists studying them, that name, Lactobacillus, well, it became a bit of a problem, like a really big, messy umbrella covering way too many different things.
That's a perfect way to put it. I mean, the sheer variety crammed under that one name made it incredibly difficult to figure out which bug did what, who was related to whom, their specific jobs. It was actually holding back the science.
So the deep dive we're doing today focuses on this landmark study that basically said, okay, time to fix this. They used the latest genetic tools to bring some order to all that chaos.
Yep. And our mission here is to unpack why that old system was broken, how scientists used these cutting-edge techniques to sort it all out, and what these new, much more precise names mean for, well, everything from research labs to your next slice of cheese.
Okay, so where do we start? The history seems important here.
Definitely. Understanding the problem shows why the solution was, frankly, so needed. So originally, classifying these bacteria was pretty straightforward, based on what scientists could see and measure.
Like their shape, what they ate.
Exactly. Their morphology, how they grew, the sugars they fermented, the byproducts they made, what we call phenotypic traits, based on what they did.
Makes sense. But science moved on.
It did. In the 20th century, genetics came in. Methods like comparing how much DNA hybridized or the GC content, and then the big one, using the 16S rRNA gene.
Ah, yes, the 16S gene. That's like the universal barcode for bacteria, isn't it?
Yeah.
Used for building evolutionary trees.
It was a huge step forward for many bacterial groups, absolutely. But as the source material points out, for lactobacilli, it just wasn't quite enough.
Not enough detail.
Exactly. It didn't have the resolution needed. It couldn't clearly separate the increasingly huge number of species being discovered and dumped into the Lactobacillus genus. It just kept growing and growing. The paper says over 270 species by 2020.
Wow. 270 species all under one name. That sounds unwieldy. Less like a genus, more like a phone book.
Pretty much. The genetic diversity, the physiological differences within that single genus became enormous, way bigger than you'd expect for a typical bacterial genus, maybe even more than some whole families.
And that matters because
Because lumping such different organisms together makes it really hard to study them properly. Their specific ecology, how they evolved, their practical uses, you couldn't see the patterns within smaller, genuinely related groups. It masked important shared traits.
okay, so the old system based on 16S just couldn't cope with the diversity. What changed? What was the big technological leap?
The game changer was whole genome sequencing, WGS, becoming widely available and affordable. Suddenly, instead of just one gene, scientists could read the entire genetic blueprint.
The whole instruction manual, not just the index. How do you compare whole genomes to see who's related?
You use some pretty clever computational tools. For defining species, the gold standard now is ANI average nucleotide identity. If two genomes share, say, over 95% identity in their core regions, they're likely the same species.
Okay, ANI for species. What about higher up, like for genera?
For that, they use things like AAI, average amino acid identity, and especially core gene AAI, or cAAI. This looks at the similarity of essential proteins coded by genes found across the whole group. It gives a much deeper view of evolutionary distance than just 16S.
Got it. And this big study we're looking at used a polyphasic approach. Sounds fancy.
It just means they didn't bet everything on one horse. They combined multiple lines of evidence. They used ANI, AAI, cAAI. They built these really detailed evolutionary trees based on hundreds of core genes.
So comparing lots of essential genes all at once.
Right. And they also looked for specific signature genes found only in certain groups, and they brought back in the classic data metabolism, where these bugs live, their ecology. It's about building a really robust case from all angles.
And they needed a lot of data for that, I assume.
Oh, yeah. The paper mentions analyzing nearly 2,500 high-quality genomes, 2,459 to be exact, a massive computational effort using sophisticated bioinformatics tools.
Okay, so they threw the whole genomic toolkit at this huge data set. What did they find? What's the big shakeup?
This is where it gets really fascinating. The analysis, looking at the core genome trees and all the other metrics, consistently revealed 26 distinct, really well-supported clusters within that old Lactobacillus group and its close relatives.
26. So that one giant name really was hiding a lot of different groups.
Totally. And these 26 distinct phylogenetic groups became the basis for the new genera. So the outcome is huge. The original genus Lactobacillus gets amended, basically. It's shrunk down to just the group containing the original type species, Lactobacillus delbrueckii.
So Lactobacillus still exists, but it's much smaller, more specific.
Exactly. Pediococcus mostly stayed as it was. Paralactobacillus was also redefined. And then, drumroll, 23 brand new genera were created.
23 new genera, wow. All carved out of the old Lactobacillus.
Yep, filled with species that used to just be called Lactobacillus something or other. It's a major redrawing of the map.
Did this analysis reveal anything else maybe about how the broader families are related?
It did. It showed pretty clearly that another family, the Leuconostocaceae, which includes bugs like Leuconostoc and Vicella, often found with Lactobacilli, is actually nested within the Lactobacillosae family tree.
So they're not separate branches. One grew out of the other.
Essentially, yes.
Yeah.
So the proposal is to merge them, making Leuconostocaceae basically a synonym of Lactobacillaceae, streamlining at the family level, too.
And what about those older ways of classifying them? Like how they ferment sugars homofermentative versus heterofermentative. Did the genetics line up with that?
It really did. It strongly confirmed earlier ideas. Those two main fermentation types align beautifully with the new phylogenetic groups. Actually, the heterofermentative ones. Genetically, they're much closer relatives to Leuconostoc and Vicella than they are to the newly defined core Lactobacillus genus.
So their metabolism reflects deep evolutionary history. That makes sense. When they decided where to draw the lines for these 26 genera, what were the main criteria?
They had a clear checklist. First, each group had to be monophyletic, all members descending from one common ancestor and including all descendants. Then they looked for clear separation based on those genome identity values, AAI and cAAI.
Okay, evolutionary distinctness and genomic distance.
Right. They also prioritized groups where the species shared common ecological traits or physiology or maybe specific signature genes. And importantly, they tried to ensure consistency with solid previous studies where possible. It wasn't just about splitting, it was about finding biologically meaningful groups.
That sounds like a really thorough, well-reasoned approach. So let's explore this new microbial neighborhood. Can you give us some examples of these new genera and what makes them special?
Yeah, this is the cool part. The new names often reflect what these bacteria do or where they live, which was kind of lost before.
Let's start with the original club then, the slimmed-down Lactobacillus. What's in there now?
Still a very important group. It's got the type species L. delbrueckii, famous from yogurt and cheese, but also found in pigs. This group is generally homofermentative, and many are adapted to live with hosts, vertebrates mainly, but also social bees.
So familiar names like L. acidophilus from our gut or L. crispatus from the vaginal microbiome, they're still in Lactobacillus.
They are. They fit that host-adapted profile. And the source lists others from chickens, L. gallinarum, hamsters, even species found specifically in bobwhite quail, wild mice, parrots, even Himalayan marmots. So it's smaller, but still diverse in terms of the hosts it associates with.
Okay. How about genera named for a specific skill?
Great example. Amylolactobacillus. The name says it all, starch-loving lactobacillus. These guys, like the former L. amyloidicus, are homofermentative and make enzymes to digest starch outside the cell.
Starch digesters. Anything else special about them?
Yeah, they like it hot. They grow best around 45, 48 degrees Celsius, some even up to 52. So you find them in places with starch and warmth, like swine gut, sourdough sometimes, or cattle waste fermentations.
Heat-loving starch munchers. Got it.
Yeah.
What about insects? You mentioned bees earlier.
Right. There are whole genera specialized for insects now. Bombilactobacillus includes species like bee bomby, first found in bumblebees, as the name suggests.
And there's another one.
Apelactobacillus. Think apis, the honeybee genus. Species like A. kunki and A. apinorum are mainly found in honeybees and on flowers they visit.
Ah, kunki. I think the source mentioned it was first found in wine that wasn't fermenting well.
That's right, an interesting detour. But its main ecological niche really seems to be bees and flowers. And the names often honor bee researchers, which is a nice touch. The new classification really clarifies their primary habitat.
Okay, bees covered. What about the ones we find most often in fermented foods or on plants?
That's where some of the biggest new genera come in, like Campanilactobacillus and Lactoplantobacillus. These are your typical residents of fermented veggies, pickles, sauerkraut, kimchi, also fruits, sourdough, some sausages, dairy products, even beer spoilers sometimes.
Lactobacillus sounds familiar. Is that where L. plantarum ended up?
Exactly, the old L. plantarum group. And L. plantarum itself is famous, found almost everywhere. Plants, fermented foods, diverse animal guts, including humans. It's incredibly adaptable. But even within this genus, you see specialization, like L. fabifermentans for fermenting beans, or L. plagiome from fermented fish in Thailand.
And Capenolactobacillus.
Also very common in many types of fermentations, includes species like C. crustorum found in baked goods. And get this, one species, C. metriopterae, was isolated from a grasshopper.
A grasshopper. Okay, these things really get around. Is there a genus for, like, liquid environments?
There is. Lactobacillus. Pretty descriptive name, right? This group, which was part of the old L. salivarius group, is often found in wine, cider, cocoa fermentations, water kefirs, fruit juices, beer, liquor mashes, basically wet fermenting places.
Any standout species there?
Sure. L. molly is known for degrading malic acid, important in wine and cider. L. hordeae comes from barley and makes bacteriocins, which are like natural antibiotics. L. vinii has a unique way it metabolizes certain sugars.
You mentioned the old L. salivarius group. Part of it became Lactobacillus. What about the rest?
Another significant chunk became Lactobacillus. This genus is really defined by its association with hosts. You find these species commonly in the mouth and intestines of humans, cats, dogs, pigs, horses, chickens, mice, a whole range of animals.
So, strongly host-associated. Any weird examples there? The source seemed to have some fun ones.
Oh, yeah. Besides the usual suspects, there's L. acidopisces, named because it was found in sour fish. L. agilis is dominant in pigeon crops, but also turns up in sewage. And the real kicker, L. seti, isolated from the lungs of whales.
Whale lungs. Seriously. Okay, that's definitely host-associated in an unexpected way. Mind blown. What about Lactobacillus?
That was the old L. reederi group, another one with many species adapted to vertebrate guts, humans, rodents, birds, pigs. But unlike Lactobacillus, they're also commonly found in fermented cereals, plants, manure, sewage. L. fermentum is kind of the odd one out here, not strictly host-adapted.
And the names give clues again.
Sometimes. L. sicilifolus means rye-loving. L. fermenti is linked to rye bran in animal guts. L. equigenerosi was found in thoroughbred horses and is noted for being acid-tolerant and liking warmth. L. panus from sourdough and swine gut.
So we have host specialists, food specialists. What about bacteria that live where easy sugars are scarce?
Good question. That niche seems to be filled by genera like Secundolactobacillus and Polyphasic. These came from the old L. collonoids and L. vaccinosturcus groups. They're adapted to places like silage, spoiled beer, fruit mashes, where simple sugars might be used up.
So they eat other things.
They often prefer pentose sugars, for example, and might even lack enzymes for breaking down common sugars like mannitol. Secundolactobacillus mellifermentans is a classic example from spoiled beer. Fun fact, Secundolactobacillus collonoids colonies were described as hill-shaped.
Huh, okay. And any slowpokes in the group?
Yep, that would be Lentilactobacillus, the old L. buchneri group. Lenti meaning slow. Found in silage, spoiled beer, grain mashes, fermented plants. They're often characterized by slow growth on certain food sources like lactate. Includes important species like L. buchneri itself, key in silage, and L. hilgardi, both named after microbiology pioneers.
It's amazing how the genetics reveals these ecological and metabolic clusters so clearly. The paper mentions quite a few other new genera too, right?
Oh, absolutely. There's Lactobacillaceae, which holds the very diverse L. casei group found in cheese, but also many other places. Lactobacillus from steak and meat, associated with the L. sacchae group. Logolactobacillus from silage, Agrolactobacillus from compost, Schleifurolactobacillus often from spoiled drinks, Furfurolactobacillus noted for short cells, Levilactobacillus, which includes the commercially vital L. brevis, and Holzaphilia, found in flowers and named after a prominent researcher.
So many new names to learn.
It is, but it reflects the true diversity. And the source does note that even within some of these new genera, like Lactobacillus or Lactobacillus, there's still a lot of variety. It suggests ongoing adaptation, maybe swapping genes, letting them specialize in slightly different microhabitats.
Okay, so we've journeyed from this one big, messy Lactobacillus category to this much richer landscape of 26 distinct genera, each with its own kind of lifestyle and evolutionary path. The big question then, why does this matter to, someone listening right now? What's the so what?
Well, fundamentally, it brings clarity, much needed clarity to a super important group of bacteria. For scientists, it's huge. It gives them a stable, reliable framework based on evolution. They can finally study these groups properly, their physiology, ecology, how they evolved, knowing they're looking at truly related organisms. It allows for much more focused research.
Right, better science. What about outside the lab?
The practical impact is potentially enormous.
Yeah.
Think about food production making, yogurt, cheese, sauerkraut. Think about animal feed additives. And maybe most relevant for many listeners, probiotics.
Ah, yes, probiotics.
Exactly. Instead of just saying contains lactobacillus, which was always a bit vague given the diversity, companies and researchers can now identify precisely which species from which specific new genus might have a particular health benefit or perform a specific function in fermentation.
So it's about precision, matching the right bug to the right job, or the right gut condition.
Precisely that. It helps refine existing applications and discover new ones because we actually understand who's who and what they're likely capable of based on their relatives and their genomic toolkit. No more ambiguity.
So even though we might still see Lactobacillus acidophilus on a yogurt pot for a while, the underlying science is now much more solid. Can we still casually call them lactobacilli?
Oh, absolutely. The paper even says the general term lactobacilli is fine for everyday conversation, referring to the whole collection of these related bugs. It's the formal scientific names that needed the update for accuracy in research, regulation, and industry. Plus, this new system makes it way easier to classify new species correctly when they're discovered.
Okay, that makes sense. So this deep dive has really shown how powerful modern genomics is. It took this confusing century-old category and revealed this incredibly detailed map of 26 distinct microbial lineages.
Yeah, it's a beautiful example of how technology drives discovery and reshapes our understanding of the natural world, even at the microscopic level.
And here's a final thought the paper sparked for me. The authors point out that most of the species we know about, the ones classified in this study, came from humans, domesticated animals, and the foods we make.
Right, places we look often.
Exactly. But think about the vastness of the planet. Wild plants, soil, water, the guts of countless wild animals that haven't really been studied for their microbes.
You're thinking there could be way more out there.
Totally. These 26 genera, as big a leap as this reclassification is, would just be the tip of the iceberg. What other amazing lactobacilli diversity is hiding out there in environments we haven't even scratched the surface of?
That's a great point. There's likely a huge undiscovered world of these bacteria out there.
So next time you grab that yogurt or bite into a pickle, maybe give a little nod to the incredible newly mapped complexity of the microbes doing the work and wonder what other microbial secrets are still waiting to be uncovered.